I'm very happy to welcome here Dr. Liliana D'Alba. So she's an evolutionary biologist and holds a permanent scientific research position in the Naturalist Biodiversity Center. And she's also a guest professor at Ghent University. Before that, she also was a researcher and a guest professor in University of Akron. And well, during her career, she has explored the function, mechanism, and evolution of bird coloration and avian and reptile eggs coloration. She has published over 80 papers on the topic, and including papers at Nature, SIGGRAPH, Proceedings of Natural American Science Foundation, all that thing. So in her talk today, she will cover some of the physical, chemical, and biological foundations of avian coloration and discuss how recent discoveries are reshaping our understanding of its evolution and function. We think this is a very fascinating topic, and we hope we get some inspiration so we can continue pushing how we understand and how we model this very complex appearance in the real world. So, all that said, thanks a lot for coming and for accepting our invitation, and yeah, the floor is yours.
Thank you. Can everyone hear me well? Yes? Thank you for that nice introduction, and thank you very much for the invitation. This is really exciting to me. My passion is color science, the biology of color, and I hope that at the end of this talk I give you a good image of what is it that I do and why I love it so much. So, yes, so this is roughly the schedule for today, what I have. Without going into too much detail, what I want is to transmit this passion for this topic. So it all started for me when I worked as a biology student, observing seabirds in their natural habitat. And then, for me, a big source of inspiration was Nico Timbergen, who was a biologist and a Nobel Prize winner from the Netherlands. And he established the field of animal behavior, or ethology, starting with a single, very simple problem. Why is it that gulls have that red dot in their bill? That was his question. And with that simple question, he demonstrated that this is a very, very important innate stimulus that starts a whole behavior of pecking. So the chicks peck this red dot on their parents and make the parent vomit, regurgitate the food for the chick. So this red dot is a very strong stimulus for the chick, but it also signals something else. It signals the capacity of this bird, this parent, to forage, to acquire the resources that they need to feed the chick, because the color that's produced on the bill depends on of carotenoids, the pigments that they are absorbing from all the fish, the crustaceans, the shrimp that they are eating. So color, you can see, it's very important for communication between the parent and the offspring, and it's a key component on the acquisition of resources by the chicks. Now, if you think of another species, right, another aspect of color in this same system of seabirds. So seabirds have different colors, and sometimes there are subtle differences, but they are very important because in this case, for example, the hues, the different shades of gray, blacks that they have on their back or the color of their feet will tell you which species is which. And this is very important because animals do not want to breed with the wrong species. So this aspect of coloration in these birds, it's very important for species recognition. But of course, colors and individuals do not occur in isolation. They are part of an environment which is very important for the expression and the perception of these colors. So in this case, when you put the chick into its context, it's very becomes obvious that the coloration patterns of the chicks help him be camouflaged, blend with its environment, so in order to avoid predation and die. And Also, we have been studying many other functions of different aspects of coloration of these animals, and we have demonstrated more or less recently that the amount of pigments, in this case melanin, that is deposited in these feathers increases the strength of the feathers themselves. And very recently, one of my former PhD students demonstrated that not only that, this deposition of pigment, which absorbs solar radiation very strongly, has an effect in reducing drag during flight. So it makes actually flight more efficient. All right, so these are some of the functions that we are working on, but it's clear that color in animal coloration, it really offers a window into the interaction between organisms and their environment. And this is fascinating to me. So we have, for example, that black vulture over there is completely black. And that black skin, that dark skin, as I said, it protects from solar radiation. Many birds have dark skin, and those birds that have dark skin happen to live in areas of the world that receive very strong ultraviolet radiation. I don't know if you spotted the bird that's occurring present in the second photo is perfectly camouflaged, pretending to be a dead trunk. That's a botu. So that's a very essential part of a function of coloration. And of course, we have the opposite when these animals want to be showy in order to get mates. But that happens not only in birds. This is a really wonderful, colorful world, and this is what's driving my research. I want to know why and what explains this remarkable diversity and colors across the animal world. So this is diversity in phenotypic. So one of the first questions that I ask, I'm a very mechanistic biologist. I want to know how these colors are produced. So we are going to review some of the main mechanisms. And I'm going to use, for the most part, examples that I have learned in birds, because that's my main working system. But of course, as I showed, many of these mechanisms applies to many other animals. So we have those colorations, those colors that depend on the deposition of pigments. And in animals, some of the main common pigments around are carotenoids, the same ones that you have that you eat in your carrots. So if you listen to your mom, she was right. Eat your carrots. They are very good for you because they have antioxidants, apart from giving this bright reddish or oranges or even yellow coloration. So the color production then depends on the absorption of light by the molecule, the pigment molecule, and the intensity of the colors depends on how much of this pigment is put into the tissues, into the feathers, the skin. Another family of pigments that are very important, and this is one of the foci of my research, is melanin pigments. They are very important and they have many functions. Though those colors, those melanins, is a group of or a family of pigments. They mostly color animals drab colors, blacks, browns, slightly reddish, but they become very important in the next slides. Then we have one of my main interests, structural coloration. That is produced not through pigments, but through the structures themselves that make the feathers or the skin. And these structures that are at nanoscale, they are very tiny structures that are organized and interfere with light in a constructive way, so that wavelengths that are in phase will become reinforced, and then depending on the specific wavelength, the size of the wavelength, that determines the color that we see that's produced. So that's kind of like a drawing that we produced of a cut of inside of one of the little branches of a feather that's called a barb. And the spongy type of structure that you see there and I should point here, but should change to my glasses, shows where those nanostructures are. This is made of keratin, the same material that's making the entire feather. It's just that at the core, it becomes organized, making like a sponge type of structure at nanometric scale. And this interferes with light, create these interfaces that are separated very regularly, and that creates constructive wavelengths. And then it's my favorite. This type of coloration is also structural, but unlike the previous one, this changes hue depending on the angle at which is seen. And it creates some of the most colorful examples that we see in nature. It's very striking. It also depends then on the interaction with light and nanostructures that become organized. But there are many more physical mechanisms that we have, for example, diffraction gratings, thin films, really photonic structures. It's a whole world of nanostructures that produce these types of colors. One of my favorite examples of this type of color is, of course, the hummingbird that are native to my native Mexico. This you can see very clearly. It changes coloration from black, basically, to very bright Mexican pink. And it is remarkable. So here, you can see that if you were to zoom in to look at any of these feathers from the gorget through the microscope, and you zoom in, you can see where these structures are. So inside the smallest branches of the feathers, called the barbules, are organelles, very tiny particles that contain melanin. And these melanosomes, these organelles called melanosomes, become stacked, right? Some of these particles contain melanin, but they also are hollow. They have some little holes. And then what happens is that they become a stack, and you have three materials that are interacting with light. You have melanin, you have air, and you have the keratin of the feather. And light has to travel through all these. And because all these materials have different refractive indices, you get a whole world of photonic effects. So that is, in brief, what happens there. Of course, these types of nanostructures are not exclusive to birds. We have examples of photonic structures that produce different kinds of structural colorations throughout animals. These are some examples coming out from our lab. And if you like plants, I was very surprised to learn a few years ago that this plant called Begonia pavonina, pavo like in Turkey, it's iridescent, and it has a very, very similar example of multi-layer organelles inside the leaves that produce this iridescence. You might be very familiar with nacre, this opalescent effect created by the stacking of the calcium carbonate that becomes organized in between organic material. And very recently, another PhD student, Jessica Dobson, she documented the occurrence of iridescence in mammal hair, because previously we thought iridescence was across all vertebrates except on mammals. And that's not the case. This is a golden ball, and she found it in other rodents and related mammals. So their hairs are iridescent, but these are moles. These live underground. They do not have, well, they do have eyes, but they are blind. So they look very, very nice, but they cannot never see each other or appreciate the beauty of the fur. But nevertheless, the physical mechanism is there. And of course, it has other functions that are related to the shape of the flattening and the smoothing of their hairs that are needed for them to travel through sand or the substrate underground. So going back to the melanosomes, these packets, these organelles that are full of melanin, in the past we have started building a database of this shape, the size and shape of these melanosomes in bird plumage. Because In humans, for example, and in mammals, these organelles they either look like sausages, very long, and in that case, they produce very dark, like black coloration or dark brown. But sometimes, like in people with red hair, naturally red, the melanosomes are very round and contain a different kind of melanin, so different chemistry that produces very reddish coloration. And that's it. But in birds, they exploded this diversity of shapes. And the shape, the geometry, the size of these melanosomes is correlated with the colors they produce. So that is very interesting, and it was a very nice project over a decade ago, and that was it. Until one of our colleagues, Jakob Winter, back then finishing his PhD at Yale University, he's a paleontologist, and he found this feather that he looked under a microscope, a scanning electron microscope, and saw that in the areas where you can visibly see that the feather is pigmented, he found these particles that people used to, or other paleontologists used to interpret as being bacteria, but they were absent in the areas that do not seem pigmented. So he was really wondering, why is this bacteria only basically exclusively present in these dark areas? We had contact with him back then, and so he knew of our work, and so that's when this wonderful collaboration started that allows us to look at first of all, we showed that was not bacteria, that those are melanosomes, and melanin is a pigment that's very resistant. It's a polymer, a very long polymer, is very resistant, and it fossilizes. So if you have a good fossil that is pristine, that has a very good level of preservation, like feathered dinosaurs in China, coming from China, these are wonderful. It's a wonderful resource that we have been using over the last years to scan for the presence of these melanosomes, because now we have the knowledge with this database of this association between melanosome size and shape and the color they produce. So the obvious question is, can we do the same with ancestors of birds? And the answer was yes. If you look, this is a fossil that is, or the first fossil that we worked on, a theropod dinosaur, chicken size, very cute. And we sampled it in different locations where the feathers are clearly preserved, all over basically the body. It was remarkable preservation. And with that, we had, this is the database of this association, shape, size, shape, and color, that distinguishes the different colors that can be produced by these melanosomes. If we plot the data from the fossil into this existing or living bird morphology, what we were able to do is actually paint, reconstruct, make predictions for what color this dinosaur would have. And this was the result. So that was the first example of this work. It was very exciting because, as you can see, it has some color patterns that we recognize in living birds. It has a red crest. We see that in birds like woodpeckers, and we are learning what the function of that is. We see the spangling, the banding on the wings, and we have very good evidence for what the function of that type of color pattern is in chickens, for example, in many other birds. And over the years, we have done this as more fossils have become available. Microraptor, Keihong, more recently a pterosaur. And this has been very exciting because what we have been able to predict, to actually say something about what the real colors of some of these dinosaurs, feathered dinosaurs, were, is starting to permeate into the media, into the public knowledge in a very nicely way. This is a very recent documentary, Life on Our Planet, where I wasn't expecting it, and then I saw, there's Anchiornis, and I was very happy to see that I was at least, I helped with this representation, to advance the representation and our knowledge about how these animals, extinct animals, look like. This is an example also from the iridescent microraptor. This was a little bit exaggerated, but it looks beautiful. So very happy with that. Now, what we have learned with these examples of feathered dinosaurs, why is it important to know what colors they wear? Well, in this case, what you see here is a tree, right, showing the relationships of the different types of animals, vertebrates, including mammals, archosaurs, dinosaurs, etc. In the upper part, it shows this plot of the size, the shape of the melanosomes, and where the main diversity is or not. We are limited, like in what we can do or what we can say, the level of prediction we have for colors for the different groups. For mammals, for reptiles, there's really not much variation of the shape, so we cannot confidently make any color predictions. But in many, the lineage of the birds, the dinosaurs, theropod dinosaurs, we can, because there's this similar diversity, and it fits quite well. So the take-home message after all these studies on dinosaurs is that we see coloration in feathers well before these animals were able to fly, to use their feathers for powered flight. So feathers were used for maybe similar functions like communication, signaling, camouflage, before they used them for powered flight. So that was a very nice insight that we got. And we keep working on this area, so keep an eye for another maybe paper in you these days. Now, in the second part of the talk, I'm gonna talk about two examples of recent projects that we've had, and my hope is that it will give you a better idea of what is the approaches that we use to do in this type of research. Mostly as biologists, we also collaborate, as I mentioned, with paleontologists, but we also work with physicists and chemists because this is a very integrative science. We do a lot of morphology. We measure a lot of structures at different scales, so therefore we use microscopy. We use a lot of traditional histology, and more recently we have been starting to add optical simulations and visual models, because what we want to know, sometimes we cannot make experiments with simple descriptions of how the colors are produced. And optical simulations allow us, it gives us an arena where we can vary different parameters of morphology and then we can make predictions of what colors can be produced and maybe why birds, for example, are incapable of producing certain other colors. And visual models because we also want to know how are these colors perceived by the viewer in the context of different environments. So that is what we are doing. You perhaps have seen this bird in this very common Eurasian coot. They are coots, I think, because they make this call that sounds like coot, coot, coot. So if you're walking in Amsterdam one day or canals in Belgium, you will see them everywhere. I don't know if you can see them here, but all they need is a body of water. Well, for many, many years, I wonder what's going on with this shield, the frontal shield. It was very striking to me as I saw them, because sometimes it's so bright. It's one of the brightest structures that I've seen in birds. Sometimes it's so bright that it seemed to me like it had the appearance of glowing. That's kind of bluish glow. And so I really wanted to know, how is this made? Why do they have it? So this is a spectral curve showing the reflectance of the different parts of the body. The two very light curves up on their upper lines show the measurement, the reflectance of the shield itself in two different methods, like measuring the specular, and I apologize, it's very, very light. So the grayish line, can you see the difference? So the grayish line is showing the diffuse reflectance. This is a shield that is not glossy, it's completely matte, very, very bright and very matte. So that's also unusual. And the other measurement is the specular reflectance. So these shields are really unusually bright, but only in diffuse reflectance. And they create a very big contrast, a very large contrast, obviously, with their plumage, which we can visually see. But for us, it's also important to look at these metrics in the ultraviolet light, because, of course, I haven't mentioned, but it's really important for many animals other than mammals that birds can perceive ultraviolet light. So we don't always know what the UV is doing, but we know in some cases it's really important. And creating contrast also in that channel is very important for communication, for example. Yeah, so diffuse, there it was. Diffuse and specular reflectance. We know that the males have larger shields and they grow them just before the breeding season. So this hints as being an ornamental part of the body they use to advertise how powerful, how strong they are to other males. And if you're ever curious, just calm down and see one of these fights between male coots. Man, they are really aggressive. And the good thing is that they advertise their shields to avoid physical contact. They are really just like, look at my shield, I'm gonna win. Don't even mess around. And that's what they're doing with their shields. So it's very important to have a very large, super bright shield. What you see here is kind of like a primer for the histology, what's happening. So this is a skin. The shield is made of skin. Imagine ultra bright skin that looks a little bit like a sponge. So that's a drawing just to orient you a little bit. And the two images in blue are showing two different types of what is really a gradient of morphology. Some of these shields, when you cut them, look like this, very porous. The other ones, very flat. So what I wanted to look at is all the holes in that patch of skin. So for this study, we use optical simulations using the finite domain time difference method, using a commercial solver that uses Maxwell equations so we can simulate how electromagnetism interact with the structures that we want to model. So we create a 3D model or 2D model that can be used both ways of the structures that we are interested in. So that is one of the models that is based on the micrograph, the electron microscopy image that we got for the skin that I just showed you. This is an experimental, basically, what happens if our hypothesis is that is the holes that are creating or increasing the scattering of light, then you can actually manipulate the number of holes in a realistic way, make some experiment where you change the number of holes or porosity of the structure and see what the result is. And that's exactly what we did. And you can see here, so you will see how light is traveling, propagating through the structure, and maybe you can see the intensity of light that's being scattered. I'm not sure if you can see, but in the light, in the left panel, you can actually see that the light actually starts being reflected, and it keeps being reflected way after this one that is very not porous. So that's what we do, and that leads us to conclude that is indeed the level of porosity of this skin that is increasing and scattering in these shields. Okay. So we have other examples of how animals and other birds use these ultra-white feathers or patches of their body to signal. This is another very interesting bird called the Eurasian woodcock. It's a nocturnal animal. You would not see it. It's very difficult to see because it's almost perfectly camouflaged. And it only is active at night. But it has under the tail feathers, the tips are white, are very, very bright white. And it only displays it during courtship, sticking the butt up, basically sticking the feathers up to present to the female, or when it's flying. And so this is another example of a structure that uses a structural organization of the tissues to increase the reflectance to act as a very powerful signal. Now, I'm going to move to how do we know how these colors are perceived by other animals, either other birds, but perhaps potential predators. And for this, I'm going to use the example of this bird. It's a western bluebird, bluebill. Sorry, there are many bluebirds, bluebill, and this lives in West Africa. Look at the bill, please. I don't know if you can see that it changes actually the color as it changes the angle. It goes from deep blue to silver, sometimes black. So it's iridescent. And this had not been reported before. We thought that iridescence happens only in the plumage. So we all obviously wanted to know how this iridescence is produced. But more importantly, can the female actually see the changes in colors as the male moves? And oops, sorry. Yeah. Obviously, for this part to know how animals are perceiving colors, we need to take into consideration their perceptual sensitivities. How many photoreceptors, what kind, the abundance, all these things. And we know quite a bit about the different photoreceptors in the main groups of animals. So we know that, for example, most mammals, unlike us, have only 2 photoreceptors. So they cannot see long wavelengths, they cannot see reddish, oranges. Birds, we know they have 4 photoreceptors, including the ultraviolet, the ones that's sensitive to ultraviolet. Reptiles also have ultraviolet, etc. Some fish, like that coelacanth, that's a very ancient type of fish, it only has 1. It lives in very, very turbulent, very turbid, waters, very dark waters, so visual perception is not perhaps that important. And there are crazy animals, butterflies. And if you have heard of this, the mantis shrimp, we don't know why these animals have so many photoreceptors. And potentially, they can see colors, a palette that we can only imagine, exponentially. We don't know. Now, whether their brain can process this amount of information to make sense of that, that's a different thing. But they do have the photoreceptors. Butterflies, indeed, very complex. Okay, so we take this information into account. It's still a little bit limited. We don't have it for all animals, but we take this information into account to produce quantitative visual models. And in brief, what we do is we use our reflectance curves as a point of reference, and we perform segmentation of these reflectance in different wavelengths that are associated with the different colors. Then we map these segments into coordinates that in a color space that depends entirely on this information. Well, we have the reflectance, we have the type of illuminant. For the most part, in the past, we used traditional CA illuminants, the broad light, the D65, D50, for example. More recently, John Endler, who's a biologist and professor of sensory ecology at Keynote University, he pioneered this and built or collected a lot of measurements, irradiance measurements in different types of habitats and light conditions, and provided us with some standardized, more illuminants that we can use in these models. And of course, then we need the information of the photoreceptor sensitivity and the abundance to calculate the noise. And then we use we feed this information into a grid that calculates distances, for example, between the coordinates of 1 point or 2 points, let's say, that we are interested in, to see the minimum perceptual differences. So the distances that can lead to the JNDs, or just noticeable differences, to actually determine whether colors can be perceived or not. So you might be familiar with this, but if not, what that grid is showing, all the points that are inside the smallest ring are comparisons or pairs of coordinates that cannot be perceived. Anything under number 1, J and D, is really not perceived. Anything above is just noticeable, and the yellow triangle is a very conspicuous difference. It's something that is obviously very, very well seen. And this is just to show the importance of the illuminant. We biologists, we haven't been experimenting much, like looking at really how the environment changes the ability to perceive these colors, but it's becoming more obvious that if you vary this illuminant, the ability of another individual to perceive changes dramatically. And in this case, for this species lives in the undergrowth of forests where it's shaded. It's very shaded. And many of these structural colorations actually happen in this type of environments where they seem to be much better perceived than in very open environments. So we are trying to understand much more what is the effect of this illuminant on this type of perception. But given our measurements, we did the morphology, we did the optical modeling, and we calculated, so we measured the iridescence, the degree of the strength of the iridescence of the bill as changing the reflectance measurements with the angle. So you can see the different curves are the different angles. And we can see that the just noticeable differences between angles, they are all over 1. So this tells us, indeed, the females can see every single change in hue as the male is moving its beak. Okay, so in general, that's what we've been doing, but we are moving into a slightly different direction now, because we can continue all our lives to describing how different amazing colors are produced. But what is really needed now is to understand how are these structures developing. How do they form as the animals are developing when they are embryos? Because this actually is very interesting, not only for us biologists. It can give a better understanding that can be applied to produce materials with these optical properties. So if we can understand what are the limitations that animals have, why can they develop or not, or grow these organized structures, we can then apply that to produce materials with these properties. So we are going to be looking at developing nanostructures inside their tissues and different types of vertebrates. And so that is very exciting research to come. And another area that we've been putting our eyes on, and I'm not sure if you have been familiar, this has been in the news more or less recently, some reports of birds, but also mammals, showing fluorescence. And there's right now a debate between, okay, we know how this is produced, fluorescent tissues, but we don't know if actually animals are using it for anything, because these are only visible at night when you shine the animal with very strong ultraviolet light, which actually is what fluorescence needs to express. So we are in the middle of this debate, and we hope to add to that conversation very soon. And with that, I really need to thank my team, because most of the studies, or the results that I showed, are produced by these. Jonathan and Nikolai were former PhD students. Matt Shoki is my long-term collaborator. Jessica is the one working on mammals right now, iridescent mammals, just finishing. And Julia and Jacob are my closest paleontologist collaborators. And of course, my funding sources. And with that, I would love to initiate a discussion with you.
Okay, so we have plenty of time for questions. So, is there any question? You pick one.
Thanks for the fascinating talk. I have many questions, but I will just ask two. The first one is with the coloration. Are there studies of like how this nanostructure degrade? So, for example, if I read a paper that it was the iridescent color, they don't weather as same as the melanin, for example. And I was wondering if there is a study of how these structures change, not only how the color change, but how the structure change in this deterioration.
So that's very interesting, because actually structural colorations are much more resistant to wear than any pigmentary color. And that's the attractive aspect of this, for example, for material scientists, but also artists. That we are collaborating with them because they want to apply these physical principles to make color. Because as long as you have the structure, and these are robust structures, melanosomes, as we showed, they fossilize. They are basically present in millions of years. So as long as the structure is there, the color will be there. Of course, if the structure, an example would be morpho butterflies, right? So there are bright blue, exactly the same mechanisms, very complicated nanostructures, but they are made of chitin. The scales are made of chitin, and the nanostructures are made of chitin, and they are very fragile. If you touch a butterfly, morpho butterfly, or if it gets wet, the color will change. So how resistant it is depends on the material that's making the structure. Yeah.
Okay. Thanks. And the second question is more open. Do you think that there could be a collaboration between the graphics community, and the biology? So like graphics can create tools for the biology to test the hypothesis?
I would love to think that yes, because I would love to collaborate with any of you. I think we can benefit. I would have to have more time to think, but I was listening to some of the talks this morning, and I can see some common ground there. So the answer, I really hope is yes, but yeah.
Okay, thanks.
Thanks for the talk. Some of the photonic crystals you've shown, or many of them, have irregularities.
Sorry, are what?
They have irregularities, so they're not ideal photonic crystals. And this is usually very difficult to model optically. So I was wondering, what do you think is the impact of those irregularities on appearance, and also perhaps on fitness?
Yeah, that's a very relevant question, and different teams, including us, we're trying to get to that answer, because indeed, it's never perfect. You can use perfect structures in models, and it will give you a highly saturated, perfect iridescence, and that is not the case. But in some cases, we know that at least for the non-iridescent structural colors, that irregularity, that level of randomness that's added to the structure just because it's biology, it's important. Actually, it contributes to that. In which way? I think it will depend on the case, what level of irregularity. But it is definitely playing a role, because indeed there's perhaps two examples that I can think of that of nanostructures, natural nanostructures that approximate to perfect photonic structure, crystalline structure. There are some gyroids in beetles, actually called weevils, if anyone is interested, and those are almost perfect crystalline. But for the rest, they are very irregular, and we need to know how they are contributing. We're getting there.
Thanks.
Thank you.
Thanks a lot for the talk. It was fascinating. I have a question about what you mentioned at the beginning of the talk. You made a distinction between structural coloration and pigments, and I guess I'm not familiar with this, but I guess pigments have a nanoscale explanation for their color as well, and I was wondering where the biology stops and the chemistry starts, and like what you consider just like a fundamental source of color versus something that the animal can control, or that the evolutionary process can control. And like, yeah, I guess is asking the question, why is melanin the color that it is, also part of your research subject, or do you just take it as an input and at some point you had to say, okay, this is not part of the like space? I hope that makes sense.
Yeah, good sense. Yes, so like pigments, different classes of pigments have been very well characterized. For the most part, sometimes we have a weird pigment, but carotenoids, for example, the whole family is very well characterized. So we know the molecular structure, and we know why this chromophore is absorbing specific wavelengths. So that is kind of established, and so that's the limitation. Animals don't have any control for that. That's the molecular structure of that pigment. Now, when we come to melanin, that's a whole entire world, because depending on the animal, I just talked about the case where melanin is inside these packets, these organelles. And there is, so the shape, the size, not so much the chemistry, but the size of the organelles that will determine how they become packed and organized, and therefore the color they produce. That is what's evolving. So we have been running models that tells us what could be the ancestral shape of an organelle and how it's changing in different organisms. So we have the example of the hummingbird that I showed is the most derived, is the most different evolved shape of melanosome that we know of. And so there's a lot of selection, evolutionary selection for this change of in the shape of these organelles. So that is what is more labile. Yeah?
Yeah, that makes a lot of sense. Thank you.
Yeah, so just a remark regarding the fluorescence. Actually, fluorescence can increase the gamut of colors that you can achieve. So maybe there's something around here that is playing. So my question was more about the beak of the bird, and do you have more insights of why it's showing that well in the forest where all the light is indirect, so it's coming from everywhere, and all this directional dependency of iridescence shouldn't really happen. And in the wild, where you have a directional light, which is basically the sun, it should be the strongest, but it appears not to be. So it's very counterintuitive here.
Yeah, that's a fascinating question, because until not long ago, I used to think the same, like, okay, why these iridescent colors should benefit from being displayed in an open environment where there's plenty of light. But actually, iridescent colors are very good for camouflaging, believe it or not. And also, when we start looking at how the animals are using iridescent colors in their behavior, in their courtship, and depending when they are living, it is becoming more obvious that these iridescent colors are often used for direct close-range communication for the potential mate. So in some cases, to the extreme that they need to be at a certain angle the animal, like birds of paradise are a good example. You would think that they are completely showy, and they are, but only when strategically they are placed where they should be to capture the attention. So they will only look super bright at a very, very specific angle. Beetles, for example, many of the iridescent beetles are very strongly iridescent. They live in very dense vegetation, and there the iridescence provides very good effective camouflage with the different rays of light that get filtered through the vegetation. So I think this is where the physics, what we know about how these colors are displayed, and the biology, sometimes they need to communicate better, because we need to understand how these colors are actually used depending on the environment. Thanks.
Well, thank you for the talk. I think this is like, you could easily spend 100 years on this and still not be done, probably. It's an amazing topic. You mentioned at the end, connecting to the previous question, fluorescence, that we are finding it in nocturnal animals and not entirely sure what the point of it is. My question was, have we found it in non-nocturnal birds, the opposite of whatever the word for that is? Because there, we humans use fluorescence for all kinds of things under daylight exposure. So I could see some relevance there, perhaps.
Yeah, it is present in birds or animals that are diurnal. Like, okay, an example is chicken eggs. The brown eggs are strongly fluorescent. Do we see that fluorescence during the day? No. Put that egg in the dark and shine blue light, and it fluoresces red, really glows red. So that's a good example of many. My view on this is that because these fluorescent structures need very strong ultraviolet light to excite and basically be absorbed and then lead to the emission of visible light, it's a very artificial setup. During the daylight, of course, we have very strong ultraviolet light from the sun, but it is, compared to the visible wavelengths, it gets watered down. So the fluorescent signal is there, but it's not visible because you can see all the other visible colors. During the night, for example, moonlight. Moonlight could be a source, and this is where some of the debate happens. Then if the bird or the animal is active during the moonlight, it could be possible that some of these animals are receiving enough ultraviolet light from the moonlight, but whether this is perceived or not, that's exactly what we are trying to understand.
So you are saying that it's explicitly not an optical brightener, probably something during the day that would make it even wider, because that's probably not.
I think that goes back to that and how it's contributing to actually the colors that are seen during the day. There's a lot that I still need to understand, but given where it's occurring and how it's being used, I don't think that it has a potential to be used as a signal alone. It might contribute to the color that's visible during the day, but I am a bit skeptical to think that it has evolved as a signal. Also, many of the pigments naturally fluoresce, but the importance of the visible component overpowers the production of fluorescence.
That's very interesting. Thank you.
Thank you.
All right. So I guess one of the things that I'm curious about, is there any research on, for example, like with aliens on another planet, such as exemplified in fiction by like James Cameron's Avatar, like what kind of, are there any directions on principle, what kind of pigments or structure colors an alien could in principle have? So as opposed to just hackily giving some sort of RGB color, like a Pandora creature, could you theoretically speculate via their biochemistry on what kind of pigments they could have?
I think that coloration is a result of the chemistry, the selective forces that are, how it's being used, why is it important for the organism. Yeah, alien forms, I think the same principles would apply. There's limitations in life of what can be produced, the type of pigments that can be produced or achieved, how they can be transported to where it's needed to show. So I think the same principle, physical, chemical principles, but also how they could be used, whether they are using it for communication. Obviously, if you were talking about visual animals, then we are talking about importance of color as opposed to acoustic signals or smell. So yeah, I think it will really depend on all those limitations and opportunities for color production, expression, and perception.
Okay, and another one I have is just like, for human color perception, there's a long, medium, short cones. And of course, there are counterparts like that for different animals, like you showed. But wonder what kind of work is there and what their equivalent of like XYZ color space or RGB would be, roughly.
I'm not sure I understand your question, maybe.
What kind of color space, like example, if you were to make an image, for example, for a bird to see, what would be the equivalent of like RGB, of sRGB or XYZ that would make most sense for their perception?
Oh, to represent colors that we cannot see.
Like that like as a bird could see, for example, yes. As a bird sees it.
So one of our colleagues, Daniel, I blacked out. He's gonna be very mad at me. Daniel Hanley. Maybe last year or 2 years ago, collaborated with engineers, and they actually developed a camera that can produce images that can give us a better idea of how a bee would see a flower, or a dog would see, see us, or so. Considering all these visual systems. So I encourage you to see that, because he would be the one who, basically his work would answer perfectly the question that you have. But we are already developing those models that allows us to produce images that can give us idea of how different organisms see their world.
Hi, thanks for the great talk. When you showed the chicks, they have a totally different pigmentation. So there must be some chemical, biological process that kind of is continuous, and at some point in adulthood, it must just stop, right? Can you comment on like what kinds of things they are and what makes it stop?
Yes, I am currently working on that. One of a master student looked at basically what is the color patterns of nestlings in birds, and are there categories, what is the diversity, and why or not might be very different from the adults? And the answer is yes, they are mostly very different to the adult form, as the chick that I showed. Now there's a French researcher here, Marie Manceau. She works on developing so how the color patterns develop in birds. And she has shown, this is amazing research, she has shown that in the embryo stage, the embryos already have a map of cells that will become melanocytes, keratinocytes, and this map form like tracks, literally tracks on the doors, on their skin. And so these are not pigmented yet, it's just the differences in the types of cells. And that is at the level of how the cells are migrating, etc. But then what happens at the local level of the skin is the production or not of the different pigments locally. And whether that's where the environment, for example, environmental factors or diet, for example, will affect how much of the pigment or what kind of pigment is produced in those cells. So all birds, chicks or adults, have this map, this blueprint for how they should look. Now, the black box, what we don't know is what makes them change from the nestling stage to the adult, that the pattern changes. but it happens locally, like interaction between cells. Basically, cells are communicating to shut down or activate the production of pigments.
Awesome. Fascinating. Thank you.
Okay, yeah, we have time for a last question.
Yeah, it's more of a comment from somebody who's been working with fluorescence in computer graphics. I mean, fluorescence is unlike structure color, I would argue, because it's more of an incidental thing. It's a byproduct of chemistry and molecular structure in many cases. I mean, the floor in this room is pretty fluorescent because lignin is fluorescent, and that's not signaling. So with fluorescence, I would argue that it's, you know, the working hypothesis probably should be it's not signaling unless proven otherwise, because it just happens in so many things. There's a lot more things that are fluorescent than most people realize. You just need to, you know, purple laser and shine it at things, and if it changes color, it's fluorescent. And once you do that, you A, damage your eyesight because of the purple laser, and B, you see a lot of things change color. So
yeah. I agree with that comment. That's kind of my position, but indeed there's a big community of biologists that are really pushing for now, okay, fluorescence is the new ultraviolet. 2 decades ago, everything was ultraviolet, and it was the most important. So I agree with you. I think that type of argument opposes the idea that it has evolved for a signaling function.
Okay, so let's thanks again the speaker.